Hi everyone, welcome back. Today we're going to cover the Th17 response and the Treg response, and then at the end we'll have a nice overview of how all of these T-cell responses balance each other out. The Th17 response plays a critical role in protecting mucosal surfaces from pathogens, including bacteria, fungi, and viruses. As such, you're going to find a lot of Th17 in the gut. The Th17 cytokines drive the immune response through the recruitment and activation of neutrophils and also the production of antimicrobial molecules like defensins. In the gut, Th17 cells maintain the integrity of the epithelia by stimulating the proliferation of enterocytes and the transcription of tight junction proteins. Th17 cells are named after the key cytokine that they produce, called interleukin 17, or IL-17. It is known to induce an array of pro-inflammatory cytokines such as tumor necrosis factor, TNF-A, IL-1A, and interleukin 6. Unfortunately, this same pro-inflammatory response is also what contributes to a lot of autoimmune diseases. A cool feature of a lot of CD4 T cells is that they all experience some degree of plasticity after their initial priming. Th17 cells in particular can follow this continuum where they gradually differentiate into a Th1-Th17 hybrid, and then they become a Th1 cell with full effector functions. This plasticity is achieved through the expression of transcription factors and cytokines. It is thought that this T-cell plasticity helps to mediate pathogen removal, amplify humoral responses, and hedge against immunopathology. Here are all the cytokines and nutrients involved in Th17 plasticity. IL-12 helps to drive the differentiation of Th17 cells into their hybrid phenotype. Excess IL-12 also furthers the conversion into a full effector Th1 cell. A metabolite of vitamin A called retinoic acid plays an important role in preventing the retroconversion of Th1 back into Th17. In this way, retinoic acid is able to hedge against the autoimmune tendencies that's caused by excess Th17. On the other hand, IL-6 can reverse the polarization of Th1 back into the Th1-Th17 phenotype. Last but not least, TGF-beta plays a pivotal role, and this depends on its presence or absence. In the absence of TGF-beta, differentiation into this hybrid phenotype is enhanced. However, higher levels of TGF-beta can block and reverse the direction of the polarization. We'll revisit TGF-beta in future slides as we look at the differentiation of T regulatory cells. T regulatory cells are considered superstars in maintaining peripheral tolerance. Tregs are also known to be potent blockers of excessive immune responses and inflammation. Tregs have a wide arsenal of tools that helps to hedge against excess immune responses while increasing tolerance to autoantigens and food antigens. Here is a short list of some of Treg functions: suppressing all of these CD4 and CD8 T cells, killing CD4 T cells, suppressing the cytotoxicity of cytotoxic T cells and natural killer cells. And then they inhibit the differentiation of pro-inflammatory macrophages while increasing the polarization of anti-inflammatory macrophages. In the previous slides, we saw how TGF-beta blocked the polarization of Th17 cells into Th1 cells. Here we see the opposite, where TGF-beta is actually favoring the differentiation of T regulatory cells instead. So T regulatory cells are known to consume a large amount of this IL-2, and this actually results in the starvation of other T effector cells, otherwise known as T helper cells. So after the Tregs are activated, they begin secreting cytokines of their own that continue to suppress other T effector cells. They also secrete granzyme and perforin that induce the apoptosis of other T effector cells. You might recall from one of my previous episodes how important it is for CD4 T cells to co-stimulate a dendritic cell so that it produces more cytotoxic T cells. Well, here you see this Treg cell jumping in and blocking the process by binding with the B7 ligand instead. By modulating further binding of B7 ligands with other CD4 T cells, this Treg cell essentially inhibits the maturation of a dendritic cell and blocks the maturation and expansion of cytotoxic T cells. So increased levels of IL-10 end up suppressing other cytokines, and it helps to shift this CD8 T differentiation into the memory cell phenotype instead. This Treg response is critical for the resolution phase of an infection because 1, it protects host tissue from prolonged exposure and damage by cytotoxic T cells, and 2, it preserves populations of naive and memory CD8 T cells for future infections. So we already know that Th17 cells have this remarkable plasticity, and that's thanks to the expression of transcription factors. Well, what's cool is that polarizing cytokines can actually drive Th17 cells to become T regulatory cells with full immunosuppressive functions. This continuum also allows hybrid cells to acquire a combination of effector functions that take on characteristics of both Th17 and T regulatory cells. This continuum also allows hybrid cells to acquire a combination of effector functions from both Tregs and Th17, and that allows an immunomodulating effect. Depending on the duration of exposure to these pro-inflammatory cytokines, these Th17-like Tregs can lose all of their suppressive function and become immunopathic. When they lose all of their suppressive abilities and revert to Th17 cells, that's when the Th17 response is capable of inducing injury and autoimmune diseases. Remember how TGF-beta favors the polarization of T regulatory cells? Well, IL-6 favors the polarization of Th17 cells. This continuum can be blocked, however, by retinoic acid that inhibits the polarization of Th17 cells. In this way, retinoic acid helps protect against the immunopathology of Th17 while favoring the Treg response involved in tissue repair. The anti-inflammatory mechanisms of T regulatory cells help to balance out the pro-inflammatory responses of Th1 and Th17. However, Tregs aren't always the hero that they're envisioned to be. On one side, higher concentrations of Tregs help to increase the odds and rate of recovery, but their suppressive effects can come at the expense of increased risk of infection. For the same reason, Tregs are also recognized as a barrier to a lot of anti-tumor immunity. Over here, Th1 and Th17 come with their own mixed bag. These pro-inflammatory responses translate into decreased risk of infection, but when unchecked, can lead to increased rates of autoimmunity, cytokine storms, and even mortality. We also see a major counterbalance between the Th2 and the Th1 responses. Too much Th2 can lead to poor prognosis of infection, increased risk of atopy and allergic diseases, and increased tumor proliferation. In contrast, a strong Th1 response is necessary for neutralizing pathogens and tumor regression. However, just like too much Th17, too much Th1 can lead to tissue damage and lead to autoimmune diseases including multiple sclerosis, type 2 diabetes, rheumatoid arthritis, and Crohn's disease. After living here in Seattle for over 15 years, I wanted to pay some respect to vitamin D and its immunoregulation. So vitamin D is also a hormone, and it helps the body to, of course, absorb and retain calcium and phosphorus that's needed for bone health. But it also aids in neuromuscular function, glucose metabolism, and healthy brain cell function. Vitamin D also acts directly on tumor cells and can induce apoptosis and inhibit proliferation. In terms of regulating inflammation, a metabolite of vitamin D called 1,25D increases Treg consumption of IL-2 in the same practice that starves other T cells of IL-2 and inhibits their expansion. 1,25D also regulates the expression of the master transcription factor FOXP3, CTLA, and IL-10, while downregulating IFNG and IL-17A genes. All of this helps to drive populations of Th1, Th2, and Th17 cells to polarize into the Treg phenotype. So these are just a few reasons why vitamin D is so amazing for the immune system. For me personally, I like to go out and get vitamin D naturally and then supplement when necessary with vitamin D3. I also believe in staying active outdoors, especially during the winter, because even if your vitamin D levels get a little bit depleted in the serum, you're still getting all the benefits of those endorphins and BDNF, which also helps with learning, by the way. I hope you enjoyed this learning experience. If you liked this episode, feel free to follow along if you want to stay posted with future videos. I have one more video I'll be posting on the microbiota and autoimmunity, and then I'll be diving into other topics like exercise or the environment and food manufacturing practices, just to name a few. Thanks for joining me, and I look forward to seeing you on the next one.